All right, so lab number 3, talking about the bacteria and very, very diverse group. We'll talk about then the symbiotic theory a little bit later, but what you will need to understand that the bacteria are probably the first group of the organisms which started the photosynthesis. Photosynthesis is extremely important for all life on Earth. And then, of course, with cellular respiration. As a matter of fact, this is my most favorite reactions. It's not really favorite reactions from my chemistry background, but this is the question which you will see pretty much every single time, or almost every single time, on a lecture or in a lab tests. Okay, and the question will be, I'm paraphrasing, but write complete and balanced reaction of photosynthesis and cellular respiration, because 2 of those reactions essentially maintain or support the whole entire life on Earth, or 99% of it. So in order to do that, of course, you remember the photosynthesis and cellular respiration reactions on top of your head from Bio 1. Yes, I believe so. But just in case if you forgot, let me kind of remind you what it is. And this is what I will be looking when I will be checking your wonderful lecture as well as the lab test. Photosynthesis starts with carbon dioxide, combining with what? Water, H2O. So far, so good. Do we need anything else for photosynthesis to start? Yes, we do. Photos from Greek means light. So we have to have light. No light, no photosynthesis. You're forgetting that part. It's not complete, no balanced equation of photosynthesis. Light gives us energy. Essentially, what the photosynthesis does, it takes the energy of life and converting inorganic compound. Here we have an inorganic compound. Here we have the inorganic compound into the organic compound, into the food. Which food? Food for the bacteria, food for the plants, food for us, because they're making it for their own. We as the humans or animals, we are biological bullies. We're coming in and saying, hey plant, you cannot outrun me and I'll eat you. I need these things which you created for yourself. So yes, we're horrible to the plants. I'll address how horrible we are when we'll talk about the plants in unit 2. So this is the beginning of the reaction, known as the reactants. What would be the product of this particular reaction? We will have something nice and sweet, C6H12O6. That's the glucose. That's a formula for glucose, also fructose, a sugar, which can be converted into a bunch of other things. Anything else? Yes. Stuff which we're breathing, oxygen. If you write something like that, you are about halfway right. It's a complete reaction. Yes, you can put the water in there. I'm looking for the simplest equation of photosynthesis. This is it. It's not balanced yet because I have only 1 carbon right here and I have 6 carbons right there. So if I have 6 carbons right here, what do I need to put? 6 right here. Now, how many hydrogens do I have here? 2. How many hydrogens do I have here? 12. So I have to put 6 right there because 6 times 2 will give me 12, right? Now, 6 carbons, 12 hydrogens. How many oxygens total I have? I have 6 times 2, 12 oxygens and 6 more. 6 right here, so I have to have 12 more oxygens. That is the equation I'm looking for. Yes, you can make it smaller, you can do a subscript, and so on. Don't care about that. As long as you understand it takes 6 carbon dioxides combined with 6 waters and the light. Light is crucial. No light, no reaction. Will give me a glucose or any kind of other sugars. Doesn't really matter. That's a generic. There's a fructose, glucose, galactose, whichever. Doesn't matter. It gives me an organic compound and oxygen. Later on, our organic compound we eating and the oxygen we breathing. So far, so good. Any questions on that? As a matter of fact, let me do it green highlighting. So that's the photosynthesis. Complete and balanced. If you write something like that on the lab practical, on a lecture exam, you will have a half a point. Why half? Because there is a cellular respiration coming in. Cellular respiration, the reaction which supports your personal life. No cellular respiration, your cells will die. And what exactly starts the cellular respiration? Some sort of food, right? We have to have some food. We'll put it in the form of glucose, blood sugar. About 70 to 80% of the blood sugar will be breaking down in your brain. Normal, of course, not overloaded. So see, right here, that part. What else do we need to have for the cellular respiration? For cellular respiration, we have to have oxygen. Anything else? Not really, no. If you have food, you have oxygen, it will start in the mitochondria. That's, of course, happening predominantly in the chloroplast or cell membrane of the bacteria, and that happens either cell membrane of the bacteria in the mitochondria. What will happen whenever we exhaling? What are we exhaling? Most of you know that we're exhaling carbon dioxide. What else? Anything else? In the cold times, which is rare in the Houston area, you can actually see the steam coming out because we're exhaling what? Water. And usually students stop right there. They're saying, well, that's it. That's wrong. Because this is not the main products of the cellular respiration. The main product of the cellular respiration is break down sugar to make energy. If you don't want to write energy, you can write ATP, because that would be a molecule which is carrying the energy in human body or in most of the cells. Or if you don't, yeah, energy, very long to spell. So ATP, 3 letters only. If you don't want to write that, you can just put E, capital E. Either or will do. Energy, ATP, or E, but do not forget to do that. Because many of the students do like carbon dioxide and water and done. No, not done. The whole idea of that particular reaction is to provide you with energy. Same thing as the whole idea for the photosynthesis to have light. No light, no photosynthesis. And now we need to break it, we need to balance it out. How are we going to balance it out? Again, 6 carbons have to be 6 carbons. 12 hydrogens have to be 12 hydrogens. Therefore, we'll have what? 6 oxygens. It's in the reverse. If you remember one, you probably will remember the other. See? Starting with the carbon dioxide, water, and energy in form of light gives us sugar and oxygen. Here, starting with oxygen and sugar and oxygen give us carbon dioxide and water and energy. This is the second reaction which you need to memorize. Let's use it. Oops, not green. Let's make it yellow. This is what I'm looking for. This is the simplest version of that particular reactions. And you need to understand that light is important in the beginning and the energy at the end of each of those particular reactions. You have to have light in order to have photosynthesis, synthesis with the help of light. And the main product of the cellular respiration is actually energy, which builds up and using, well, I'm using it right now when I'm talking to you, moving my arms and so on. So please remember those. You will see them over and over and over again. Why are we talking about the bacteria? Because they are the one who started it. They start with the autotrophic, and over a period of billion of years, they were producing oxygen, releasing it into atmosphere. So now we have oxygen to breathe. They started it. Plants continue to do that particular trend. Very, very important for the life on Earth, for biology. That's why I will be asking about those particular reactions, and I will be basically talking about something, some sort of group of the organism, and we're talking about where are they getting the food, where are they getting the energy. All right. So now let's go into a pre-lab questions. What two domains contain prokaryotic organisms? Bacteria and archaea. All right. I'll go with the number 3 first. Autotrophic. Autos means by itself. Trophos means food. Making food on their own. The photosynthetic. Those ones are known as the cyanobacteria. Cyano, blue and green algae. They also refer to that. All right. That would be the autotrophic. What about the heterotrophic bacteria? Well, heterotrophic bacteria are any other bacteria which do not produce their own food. Hetero means different, trophos means food. They have to eat. We are heterotrophs. We're not bacteria, but we are heterotrophs. We have to consume something. We cannot just go bathe in the sun and, oh, photosynthesis, go. I'm getting my sugars right there. No, we cannot do that. Fortunately or unfortunately. Defined. Heterotrophic organisms have to consume organic matter. Example, all of you heard about E. coli, E. Escherichia. Instead of saying Escherichia, we're saying E, then dot, since it's an abbreviation, and coli, C-O-L-I. So cyanobacteria for the autotrophic, and autotrophic can produce their own organic compounds. Cyanobacteria, or look at those ones, Anthrax. Today you'll see some of them. Salmonella, you heard about those ones. Listeria, E. coli, Treponema, today you will see those ones. Gonorrhea, oh, E. coli right there. Should be actually italicized, right? Because it's the Latin name. So, heterotrophic have to consume organic compounds, E. coli, Anthrax, Salmonella, can make its own organic compounds, autotrophic organisms. How long have prokaryotes been around the Earth? I think your textbook saying 3.6, 3.8 billions with a B. One of my graduate professor in Moscow State, he believes that the bacteria were here for over 4 billion of years. He actually claimed that he has the fossil, which is a 4.2 billions of year old. So around 4 billions would be the correct answer. How long eukaryotes have been around on the planet Earth? Roughly 1 billion, give or take a few millions of years. Now, eukaryotic organelles probably evolved from endosymbiosis. Endosymbiosis, very simple. You talk about the endosymbiosis in Bio 1, just refreshing your memory, a large cell, prokaryotic, And we'll talk about that in a lecture too. But large cell engulfed, grabbed a smaller cell, also a bacteria cell, which were going through cellular respiration. It was getting sugar, was taking oxygen in, producing a lot of energy, like 32 to 35 ATP molecule per sugar molecule. So the big cell says, okay, here's the deal. You making the ATP for me, I will not eat you, I will not kill you, and I'm simplifying, of course, and I'll provide you with the sugar and protection, so no other big cell will eat you. Okay, cool. This is how essentially mitochondria came about, according to the endosymbiotic theory. And then that particular big cell, still prokaryotic, but already have engulfed one of those particular cells, engulfed the cyanobacteria. Cyanobacteria, light, changing the light into the energy of the light into the sugars, and the big cell saying, okay, I have the one which really likes sugars and you making sugars. So you make sugars, you make me ATP, and I'll be happy, and I'll be protecting both of you. So two possible semi-autonomous organelles, because they have their own DNA, are what? Chloroplasts and the mitochondria. Next time we'll talk about the endosymbiotic theory, but because it's talking about here and it will have questions, two organelles, chloroplast, mitochondria. Give an example of way bacteria can be beneficial to humans. Well, it's actually talking about this somewhere here. Well, first of all, photosynthetic bacteria producing oxygen. We're breathing the oxygen which has been produced by the bacteria. That's a beneficial right there. But also making some of the yogurt. You probably heard about lactobacillus, etc. Many of the cheeses. That's a direct benefit. We're also using bacteria now, genetically modified bacteria, to produce human insulin and so on. All right, now let's take a look at the table 1. Table 1 called stromatolites. And if you'll come to the lab, you will see a nice polished sphere with a bunch of layers in it, and the students were like, I'm not really sure what I'm looking at. I'm looking at the stromatolite, and it's a really nice sphere. I don't have it with me, but it's in the lab, in the face-to-face lab. So and they're like, do you find this in the nature? And I'm like, okay, well, that's clearly a problem. So basically what you will find in the nature is the rock, which looks like this, okay? And whenever you're looking like this, you will start seeing those kind of round thingies. That's, remember we were talking about the mats of the bacteria. This is what the bacteria would create. Now, if you saw that across and polish it, what you will find is this. You will start seeing the rock with some layers. You can see darker layer, you can see lighter layer. And then if you'll take that particular rock and cut it into a nice sphere and polish it, you will end up with a nice sphere. But this is a raw stromatolite. This is the cross section of the stromatolite, and this is the stromatolite in the in the shape of sphere. So now here it says, use the space below to draw the picture of the stromatolite. Some of you might be artistically oriented, some of you might not. I'm not grading you by the artistic representation, as long as I understand that you understand, well, as long as I can see you understand that there is some sort of rock with the layers. I'll give you even easier way to do it. With the snippet or anything like that, you're taking a picture of that and placing it right here. Make sure to label it if needed. In this particular case, it's not asking you to label it. But you can clearly see the layers, what the layers represent, why we start seeing the layers. Remember, the layers essentially is the mesh of the dead cells, strands, which capturing sediments. Different color means that they capture different sediments. It's located on a shallow, in a shallow part of the ocean, right next to the coastline. So let's say there was a rain, lots of rain, and then it's washed off a lot of dirt into that shallow part of the ocean. And those particular sediments, the dirt one has been captured. And then we're looking at the waves coming in, bringing the different sediments. So now we have a lighter one. So different sediments will give you a different colors. So what do you think caused those differences? Different sediments which has been captured. If assume that this is some earliest form of bacteria, would they have been autotrophic or heterotrophic? Heterotrophic or autotrophic? In order to be autotrophic, you already have to go through the process of what? Of photosynthesis. You have to evolve. Heterotrophic bacteria most likely were one of the first organisms. They were coming in and they were taking whatever the pre-made organic compounds. How the pre-made organic compounds are formed on Earth? Well, during the earliest ages of Earth, where we had the lots of volcanic activities, totally different atmosphere. There is violent storms, thunderstorms, etc. All of that could make some of the organic compounds. So basically, the first bacteria, when it's originated or came to Earth, they ended up with the primordial soup, the waters which were contain a high level of organic compounds. So heterotrophic bacteria start consuming it as they were reproducing. More and more bacteria were consuming stuff, and now we start seeing the shortage of the pre-made organic compounds on the planet. So some of the bacteria start switching to the autotrophic way of eating, so to speak. So in this particular case, going back to the draw a picture, if you don't really like to draw, you can print screen it. Let's see, where's my, let's see, print screen. The easiest way, here we go. Print screen, then the crop it, Control A, and then Control C, and then Control V into the lab. Well, you might want to resize or anything like that. Once again, it's my own images. I took those particular pictures. This is actually the rock. You don't really have to give me the credit. If you're going and taking picture from somewhere on the internet, make sure to use the credit from whatever the source on the internet you chose. And it have to be common license use, have to be a common use, Creative Commons. All right, but you have my permission to use those particular images if you decide not to draw. Some of the people are really good, easy with drawing, a square, couple of layers, done. Moving on. So name the type of the bacteria found in the stromatolites, and that would be right here, cyanobacteria. All right. Any questions, any problems? So here we have another example of the stromatolite. Here's another example of stromatolite, whichever you want. Now, table 2, talking about the model. There is several major structures, which in this particular case, you need to label. Speaking of labeling, speaking of answering, some of you does most of you does a wonderful job on answering the questions. However, whenever you're answering the questions, you can use a different color font, for example, red. Do the bacteria have it's much easier for me to see if you are using some sort of different color font. You don't have to go with red. You don't like red. Oh, I don't like red. I won't go with the purple. Fine with me, especially if it's bold. All right. Some people, some students, I don't know the reason, they go with like, I had students before going with like light gray, and then they going with like, and I barely can see that. I guess I have the old eyes or something like that. I'm like, what? And then I'm writing the email, please redo it in the normal colors. But some of you doing a good job on brightening it up. I can clearly see where your answers are. All right. So make sure that you're using some sort of bright colors. It helps me. It makes me happier. You can go with the blue. Perfect color right there. Really nice. Or green. Well, make sure that yellow is not a really good color because it starts blending in. You can kind of see right here. Right there, right? Yeah. O is almost impossible to see. So make sure to emphasize your answers. Now, on how exactly you're going to label the following structures. Where the plasma cell membrane. Some people going with a very easy way, and there is a draw, no, insert, and there is like smart art, and then, no, it's not smart art. What is that? You can go with the drawing tools and you can do something like that. That's relatively easy, but sometimes it's becoming really messy, especially if you have more than like 5 or 6 things to measure, and then it becomes like a network, a web spider, a web or something like that. You can go with insert the shapes. Many of the students going for that cell membrane. It's become really nice, but like, for example, cell membrane right here, right? But make sure that it's visible. So if you want to use that, make sure to use a better color, and might want to change the size of that particular thing, right? You can see the styles right here, and let's go with the bigger style, better see. Or maybe even change the color to something more drastic, like blue right here, and then you are going with, you'll go with another one, cell wall will be that particular light blue, and then so on. So, but whenever you're doing this, try out to avoid, like, for example, cell wall and going, and you decide, well, my cell wall will be right here. And then it's again becoming a spider web, and I need to figure it out what exactly is going on. All right, so other people do something like that. They'll put the boxes, they draw like a word box, a comment area. There's no text box. And they will put text box right in the image if they, well, so they can draw and they can answering like that. There is more than one way to do it, but as long as I can see it clearly, please, that's one of the most important things whenever you're labeling something. It's not only you labeling, I need to read it afterwards. So let's kind of say, where is the cell membrane? Cell membrane would be the outer layer right here, kind of brownish color or orangish, burnt orange, I guess. Now cell wall, that particular bluish, tealish color right here. Now that's the cell wall. Now capsule, outside of the cell wall. Again, kind of burnish orangish, but that's a cell capsule. Now chromosome, that purple thing right there. And the flagellum, actually it should be flagellum because it's a singular one right there. Also, those particular bluish circular or like number 8, this is the plasmids. The pinkish dots are the ribosomes. So you're coming on the lab practical, it might ask you name the structure, and it's showing in the purple thing. You can say chromosome, you can say nuclear region, you can say DNA. I'll accept either of them. Or this, or pilus. We already covered all of that in the lecture. Do the bacteria have a nucleus? No. What is the function of capsule? Protection and adherence. Adhere to the surface. You, for example, sneeze, bunch of bacteria flying around, like, I need to stick to the surface, right there. Which part of the bacteria is responsible for the movement? The flagellum, or flagella if it's plural. All right. Any questions, any problems on that? So those particular structures, DNA, ribosomes, the plasmids, cell membrane. Oh, cytoplasm. Can anybody show me the cytoplasm? That particular yellow stuff. The filling of the cell. That's a cytoplasm. So cell membrane, cell wall, capsule. Those hair-like projections are pili or pilus one, and then the flagellum or flagella. Now, we already talked about the peptidoglycans, and we already talked about the lipopolysaccharides. So in this particular case, you're looking at what? You're looking at the two different structures of the cell wall itself. Cell membrane right here and cell membrane right there are pretty much identical. Now here you're looking at the gram-positive cell wall. Why? It's predominantly made out of that particular blue stuff, which is a peptidoglycan layer. Do we have a peptidoglycan layer in the gram-negative bacteria? Yes, right here. So, yeah. Do not, please, do not use those particular pictures to copy and paste it into your labs. I'm giving them as the reference. Do not put the pre-made, the pictures from the guide into that particular, those ones already labeled. No, label it on your own. Otherwise, I will stop using them in the future. If I'll say, and I will deduct the points. It's already been pre-labeled for you. You can use it as a reference. Nothing stops you to label it properly nowadays, right? So anyway, whenever that's the spore, of course, now we're looking at two of those. Most likely that picture will be on the lab practical. So which one is a gram-positive, gram-negative? The one which has a thick layer, that particular bluish layer of peptidoglycan, is the gram-positive. It has, the gram-negative has a smaller layer of peptidoglycan. Still has it, but not as much. In addition to the peptidoglycan, it has a lipopolysaccharide. Something else, additional layer. Looks almost like the cell membrane layer. It's not a cell membrane layer, but it has the lipids and other things. And this is what lipo stands for in lipopolysaccharide. Okay, so be able to recognize gram-positive, gram-negative, that this is the cell wall, cell membrane, and what it's made out. Peptidoglycans, peptidoglycans plus lipopolysaccharide. Which cell wall has more peptidoglycan? This one, so it would be a gram-positive. Peptidoglycans a lot. Which type of the cell? Inner phospholipid, bottom only. You're looking at the bottoms. This one has a cell membrane. This one has a cell membrane. That one has a cell, kind of like a cell membrane on top as well. Lipopolysaccharides. That one doesn't. So gram-positive would be the only one which has the inner phospholipid bilayer. How would you differentiate between gram-positive and gram-negative cell wall? Different structure of the cell wall. Made out of only peptidoglycan, peptidoglycan plus something else, lipopolysaccharides. Penicillin, which is antibiotic, we talk about that in the lecture. Antibiotic attacks the peptidoglycan layer. It destroys, so cell is essentially spills up. So in this particular case, antibiotic goes and destroys that one. In this case, it goes, destroys the middle layer, but the cell will be kind of intact. It takes much longer for the lipopolysaccharide layer to be destroyed. All right. So in this particular case, do you think the penicillin will be most likely effective on a gram-negative or gram-positive bacteria? Most likely will be effective on a gram-positive bacteria. It will work right away because it's destroyed peptidoglycan layers. They have something else in addition to the peptidoglycan layer. All right. So this is the gram-positive and gram-negative. Now, the models. We already talked about that, heterotrophic. In this particular case, most likely you'll see this particular picture in a lab practical, and the arrow probably will point somewhere, and it will say name the shape. And if it's kind of pointing toward the rod shape, that would be the bacillus. Roundish, it's not a perfect round, but not a perfect sphere, but roundish, that would be a coccus. That one is the spirula or spirochete, right? And those ones, kind of sausages looking like, that would be vibrios. What is the function of endospore? Well, let's try to figure it out where the endospore is located at. See that particular rod-looking bacteria. Here we have a perfect rod, and then there is a brown thing inside. Endo, and looks like a spore. So endospore right there. That brown structure is the endospore. What about those ones? Are they nuclei? No, they're not nuclei. I had students before saying it's a nucleus. It's a bacteria. It's not, it cannot have a nucleus. If it's a nucleus, it's an endospore. Same thing right here. The white stuff is an endospore. What is the function? Protect DNA material of the bacteria from harsh environments. Too dry, not enough food, too cold, in some cases too hot. So identify coccus, bacillus, vibrio, and spirochete. So coccus, the round ones, bacillus, rod ones, vibrio, coma ones, or sausage look like, and spirochete, a corkscrew. So here we have all of them. The vibrio, kind of sausage or coma, spirochete, a corkscrew, the bacillus is a rod shape, and then the round ones would be, of course, coccus. So make sure that you know those, and you will be able to identify the endospores. So heterotrophic bacteria slides, you're coming in. I literally yesterday on Monday went into the lab to double check that the microscope's pointing out to all the bacteria. We have 5 bacteria. They all listed right here. Anthrax, Gonorrhea, Treponema, Volutanus, or Spirulina volutanus, and Vibrio Cholera. 4 out of those are gram positive. The only gram, sorry, let me go back. All of them are gram negative except the anthrax. The anthrax is the gram positive bacteria. Gonorrhea, Treponema, Volutanus, as well as the cholera would be gram negative bacteria. Now look, I had students before saying, Bacillus anthracis, what shape it is? I'm like, think. Like, about what? I'm like, it says right there, bacillus. What kind of shape is that? I don't know. I'm like, yes, you do. We just talked about that just momentarily. Come on. What's the bacillus shape? Rod. I'm like, it says already in there. Basically the rod-shaped bacteria which causes anthrax. This bacteria literally causes anthrax. Oh, I didn't think about that. Neisseria gonorrhea. Neisseria is the name so we don't really know what the shape of that is. Treponema is also, we don't know the shape is, but Spirillum volutanum, it's a spirochete right there. Like Vibrio causing cholera. So coma-shaped bacteria which cause cholera. Lots of throwing up and diarrhea. Dehydration and you're dead. Still thousands of people dying every year from that particular bacteria. Misspelled. Yes, not a very good way to die. I mean, throwing up, diarrhea, throwing up at the same time. Yeah. So, but what would be the shape? It's a Vibrio. Some of them kind of hard to see. You'll see some of the part. Look at this particular thing right here. You can see the strands. It's actually let's see if I have a bigger. No, I don't have a bigger. But it's actually a strand of the bacillus with a bacillus with a bacillus with a bacillus. That's the anthrax right there. And you see it's kind of bluish color, purplish bluish color right here. That's a gram-positive. So shape would be a rod shape. Is it a gram-positive or gram-negative? It's a gram-positive. It's a bluish, purplish, purple positive. Now, here we're looking at the gonorrhea. Yay. Well, the bacteria which cause gonorrhea. Increasing in size, you will see kind of roundish shapes right there. Now, speaking of which, let me go to anthrax. Under a higher magnification, you can see the rod-shaped structures right there, right there, right there. It's just strands of it. I had students before, like, you're looking at the gonorrhea? I'm like, I'm not looking at the gonorrhea. I'm like, why not? I don't want to get it. I'm like, you not get the gonorrhea from looking at that through the microscope. It's already been dead. I just, in case, I'm like, okay, whatever. And what about pictures? Would you look at the pictures and get the gonorrhea? I don't know. I'm like, okay, well, here, so we're looking at the clusters of the round-shaped bacteria. So Neisseria gonorrhea is coccus. And you see it's not purplish, it's kind of pinkish reddish color. That's the negative, gram-negative bacteria. On the lab practical, whatever you'll see, you will not have to guess. Let's take a look at this particular guy. Treponema cause syphilis. What kind of shape is that? Well, first of all, gram-positive or gram-negative. I already told you. The only positive we have is the anthrax. You can say it's not a reddish color. Yeah, but it's not purple either. So if it's not purple, it's negative. Now, what would be the shape? Well, like strands. Yes. Treponema is one of those particular quite interesting exceptions. First of all, it's not pinkish reddish color, even though it's negative. And second of all, that, believe it or not, is the spirochete. You can say how it's a spirillum, how it's spiral, how it's a corkscrew. It's a very, very tiny corkscrew. You can go like this. You can go. This is what the bacteria, that one is. Let me see if I have a little bit better. Oh, here you can see it right here, kind of spiraling out right there. All right. Oh, that one. You can see that one going up and down. So that's a spirillum. Yes, treponema. Gram-positive, gram-negative, not purple, therefore gram-negative. And the shape would be spirochete. Next one, Spirillum volutanum. That's a typical bacteria which you will see on a lab practical because it's a typical spirillum. If it's there, you can clearly see. It's one of the largest bacteria we can see in the nature. It does not cause any diseases or anything like that. It's actually the bacteria which lives in the soil. You can clearly see a very nice spirochete or spirillum, either or. Is it a gram-positive or gram-negative? See, pinkish color, so it's a gram-negative bacteria. Finally, Vibrio Cholera. Those ones are kind of hard to see because they're tiny, but you can kind of see sausage or small size, small kind of comma shapes right there. They're not really rods, they're curved rods, and that would be a Vibrio, which is name suggesting, right? And it's a gram-negative bacteria. All right. Now, autotrophic bacteria. Autotrophic bacteria means what? It will be doing the photosynthesis. One of the most common pigments for the photosynthesis is green, right? So if you'll see bacteria which is green, guess what? Most likely it goes through the photosynthesis because it has the chlorophyll. Not chloroplast. Make sure that you understand that. Chloroplast is the organelle, membrane bound. Those are prokaryotes. They don't have chloroplast. They have the pigments, but they don't have the organelles yet. So let's take a look at this particular guy. Actually, guys, that's a plural. That's the, again, strands. And each one of those particular lines, that's a separate cell. Now look. There is a black spot somewhere along those particular strands. Those ones are known as the separation discs. Separation disc. What does it mean? Why it's caused? Separation disc form, what causes? Death. What do you mean, death? Think about it. We have a cell, right? A kind of flattened cell. It's photosynthesized, it's producing the second one. And then more and more and more, creating this strand. And one of those cells are dying off. So they keep a colony, a strand, a filament, and then one of those particular cells dies. Whenever it dies, it's the integrity of the cell wall, cell membrane breaks down. As a result, the chemicals start getting in, and we start seeing that particular black line, separation disc. All right, so vegetative cells, all of those green ones, and then we have the black one. This is the separation disc. Now, Anabina, you already seen it in Bio 1. If you took, here we have the bigger, higher magnification. So 2 dead cells right here, 2 dead. Why it's called separation disc? If you'll take that particular filament and with the tweezers start moving in, most likely it will break at the dead cells. So here we have the answer of the separation. Now, Anabina, if you ever did the Bio 1, you already seen it. It basically will be a cluster surroundish, also photosynthetic bacteria. But here it looks pink, here it looks blue. Some of these things like, is it a gram-positive or is it gram-negative? They were using not the gram staining technique. That's why they are looked differently or slightly different. So different colors. But in this particular case, you have kind of beads of the bacteria combined together in the photosynthetic bacteria. Is it autotrophic or heterotrophic? It's a table 5 autotrophic bacteria. It's capable of photosynthesis. Is it a prokaryotic or eukaryotic? It is a bacteria. All of the bacteria are prokaryotic. If it's a bacteria, it's a prokaryote. Now, growing the bacteria, that's a kind of fun part of that particular lab. And basically, this is what we have. We have something which is known as a petri dish. Here's a single petri dish, and each group in the lab will receive one of those particular petri dishes. And then what you will need to do, you need to grow the bacteria. Well, before you will start growing the bacteria, let's kind of go over. That's a plastic dish with the top and then the bottom, and then the bottom it has that particular kind of semi-transparent gel-like material that's called agarose or nutrient agar. It's basically, remember, the bacteria needs three major things to grow. Warm, wet environment, and the food. Agarose provide the bacteria all sugars for the bacteria to grow. So whenever you labeling in the real world, whenever you're labeling the petri dishes, you're actually flipping it over and you're labeling on the bottom, not on the top. You can say, why in the world on the bottom, not on the top? Let's say you have a 10 or 15 petri dishes and you are misplace the tops. Is it possible to do? Yes. But the bottoms, you will know what exactly is growing. If you're seeding the bacteria, especially in the rush, you're seeding the bacteria, you're putting the bacteria on the agarose to grow later on. And then you're like, I don't know, was it from here or was it from there? But then you can see that the sample is from blah, blah. So usually you're marking it on the bottom. What's your name, when you seed it. So you have to put the date and possible bacteria or what you want to get the bacteria from. At 37 degrees Celsius, which is essentially a body temperature. Remember, warm, wet environment, plenty of food. So now what you will do, I will give the students those particular, well, they actually pick it up, the petri dishes. They will have something which known as the swab. And then they will need to get the bacteria, and they're like, where will get the bacteria? I'm like, anywhere. They are everywhere, literally. You need to wet the swab and then swipe something. The surface of the tables in the bacteria, well, in the lab we usually right now, well, we usually cleaning the tables with the alcohol to begin with, so they're not usually a good source for the bacteria. But the keyboards, the money, the cell phone surfaces, the hand, the bottom of your shoe, the bottom of the backpack. I had students like, can I go to the restroom? I'm like, yeah, sure, you don't even have to ask. They're like, no, can I swipe in the bathroom? I'm like, you will be surprised, our janitors do a great job. Bathrooms are quite clean, actually, in the South Campus. So they're like, but can we? I'm like, sure, yes, go ahead. So the students will use the imagination where to get the bacteria because they are everywhere. And then they will close it, they will flip it so it will prevent the evaporation, and they'll put in the incubator to incubate for 37 or so degrees, well, with 37 degrees. And let's say if we're doing it on Tuesday, on Thursday you're coming in and you'll start seeing the bacteria growth. Would it be a colony of bacteria? Remember, whenever the bacteria has plenty of food, it will start going through the binary fission. A single bacteria is impossible to see, but when it grows into millions, you start seeing the whole colony, okay? And those particular colonies you will be able to see. So this is the whole setup for the experiment. And now we start looking at the actual example, the credit card. A student pull out a credit card from the surface, from his or her wallet, I don't remember, was it a girl or boy, and they swipe it, and this is what they saw. So the credit card is not really, so here we have a colony, this is another colony. You can see that the colonies are different in shape and sizes and the colors, etc. So each one of those dots are actually represent, and that particular whole huge things was a really fast growing bacteria. Here we have bacteria, all of those, so it was completely covered. So that's the credit card. Right now let's take a look on the next one. Next one is the cell phone screen. A student swiped her cell phone screen, and each of those particular dots are colonies of the bacteria which she picked up with a swab on her cell phone. And again, remember, we are talking, we have tons of bacteria on our screen, well, on our skin, which will be transferred to our skin, and we're not really cleaning the screens too often. The next one was what it was. It was the bottom of the backpack. And this is probably some sort of fungi, that fuzzy stuff. Could be a bacteria, but probably a fungus. meaning some sort of mold, tons of spores as well. That's probably also mold, but those round shaped structures, that's a bacterial colony of different species. You can see some of them start going inside of the agarose, some of them created quite interesting things. How exactly they are seeding them? Again, going back to the petri dish, you have a swab, you swipe some sort of surface, and what you will do, very gently, you will swipe it back and forth, back and forth, back and forth, right there, kind of creating a zigzag. Then you will turn at 90 degrees and you will swipe across. So we have that one, now it goes that way. And then it's a really good idea to turn at about 45 degrees and store as much bacteria as possible. So here we have the bottom of the backpack. This one is the laptop keys. Again, each one of those dots at some point of time represented the bacteria which start growing, and now it's a colony of the bacteria growing on the agarose. That's just the laptop. And here we have a $20 bill. Again, those fuzzies, probably the fungus. Others are bacteria, tons of things right there. This one is inside of the shoe. Student pull out the shoe, swipe it inside, and as you can see, the species of the bacteria inside of the shoe are drastically different than the species of the bacteria which resides inside of the shoe. Different environments, right? Now here we have the hand. Just a person swipe the hand and move, well, created that. And now what we did is really interesting. What is that? That's the hand sample, same thing, but after the hand sanitizer has been applied. As you can see, there is like, well, maybe that one is a bacteria and that one. So not all of them has been cleaned, but definitely a much better than we had it before, right? And that's, I think, we're done with the actual lab. Let's kind of see the answer. Which surface seems to have most of the bacteria? Well, most of the bacteria, probably credit card, I think. Basically, how many dots or different colors of dots we have there. Do any of the results surprise you? Well, let me know if anything surprised you and why you were surprised. That's an open-end question. Are all of the colonies on the plate identical? Well, if you'll take a look right here, for example, they're definitely different. Some of them fuzzy, some of them round, some of them yellow, some of them white, whitish, transparent, semi-transparent, and so on. So they are different. That would be the different species. Are those bacteria autotrophic or heterotrophic? That would be a heterotrophic bacteria because they're growing on the agarose. Remember, agarose provide them with the food source. Now, post-lab questions. What part of the bacteria is affected by the Gram staining? Cell wall, specifically peptidoglycan, substance which the stain binds to peptidoglycan of the cell wall. Now, what are the four possible shapes of bacteria? Coccus, the round one, the rod-shaped bacillus, the spiral one is a spirochete or spirillum, and the comma one is the Vibrio. Name two heterotrophic bacteria. Up to you. We talk about anthrax, all of those bacteria which were anthrax, the Neisseria, Gonorrhea, Treponema, Spirillus volutanus, Vibrio Cholera, E. coli, Salmonella, all of those are heterotrophic bacteria. Name one autotrophic bacteria. We looked at the two, Oscillatoria and Anabina. Do bacteria reproduce sexually or asexually? Both. We already covered that in the lecture. Describe the condition necessary to grow bacteria in the lab. In order to grow the bacteria in the lab, you have to have what? For the bacteria to grow, you have to have three things: warm, wet environment, plenty of food. What do fossilized bacteria form? Fossilized bacteria form stromatolites. And number 8, you find a new species of bacteria under the microscope. Bacteria cells look small, bright green balls arranged in a thin line. After treating with a gram staining, the bacteria cells turn dark purple. How do you describe the bacteria? Be specific as possible. The shape, gram positive, gram negative, or anything like that. That's the up to you. All right. So make sure you will answer as specific as you can. Now, reflection questions. I have to add a few things using the complete sentences. Because I had students before, like on the evolution lab, hominid evolution, literally I'm paraphrasing, but it was like main purpose, evolution. What has been learned and observed? Evolution. One word, evolution, evolution. And how can you use, modify, and apply this concept in real life? Yes. I'm basically quoting a student, actual student paper. So in this particular case, make sure to use the complete sentences and give me like a short paragraph. I don't need the essay for 3 pages, but give me the background of what you learned, what did you do, what you observed, and can you use in the real life. If you don't think about how you can use it in real life, say just that. I don't know how exactly I can use the bacteria in real, well, the hominid evolution in real life. Well, there is quite a few things you can use it, right? You can talk to the people who thinks that we evolved from chimpanzees or monkeys, which is incorrect. So don't use that example once again, and so on. So, and we are basically done for today. It's 11:16, so we are done. If you have any questions, any problems, please let me know. I will stop the recording right now.